So my long-standing interest is genetics of bipolar disorder, and basically got into it by way of trying to understand the cause of the illness so as to develop better treatments. And genetics seemed like a powerful way to get at a fundamental understanding of the cause of the illness. More recently, though, as research moves very quickly and in sometimes unpredictable ways, stem cells have become a very interesting research tool. I'm sure you've heard this in the news. Some of you may know a lot about it. I'm learning about it, and I'll tell you some of what I'm learning. But I think that it's a very powerful tool that I'll talk about tonight that I think can help advance our understanding of the illness and lead to new approaches to treatment and medications, etc. But one of the main things I'm engaged in right now is pharmacogenetics, and that study of the genes that influence treatment response, the long-range goal of this being to have a DNA test panel so that you can get your DNA tested when you go into your doctor's office, and the results will come back and indicate whether you should best be treated with lithium, Depakote, Lamictal, etc. In other words, to give your doctor a sort of prioritized list of which medications he would be most likely to respond to. So that's part of what we're working on and part of what I'll tell you about, and part of what I'll be hitting you up for volunteers in case any of you are interested. So this is a small group. Feel free to ask questions and interrupt. We can keep it informal. I just briefly review bipolar disorder. I think this is by way of review for most of this group, but just briefly. Mania is comprised of this list of symptoms, which is most prominently a change in mood. We're accustomed to seeing that as elated mood, but what is sometimes less appreciated is how commonly that is irritable mood. And this gets in, and sometimes this is very difficult to recognize as mania when someone has a preponderance of irritable mood, or when they have what we call a mixed state, where they have symptoms of depression and mania simultaneously, which frequently is accompanied by irritable mood. Grandiosity is commonly a part of this. I feel great, I can do anything, take on anything. When people are manic, as I teach my residents and medical students over at UCSD, there's one word that almost defines it. And when somebody says it, my index of suspicion for mania goes up really high. What do you think that is? I felt close. What else? Invulnerable. Invincible. Nothing could stop me, nothing could hurt me, nothing could slow me down. They feel filled with energy, leading them to start new projects, work long hours. They will have so much energy they don't need to sleep at night, which is distinguished from depression where they're unable to sleep. They'll have an internal experience of flight of ideas or racing thoughts. This is the part of the illness that's frequently associated with creativity. And this is reflected externally with the experience. They want to tell you about all of it, so they'll talk rapidly, they'll be intrusive, they'll be difficult to interrupt, hard to get a word in edgewise. Again, with that energy, they're not simply typically frittering it away, they're on a mission, they can be very focused and directed. Unfortunately, sometimes that focus is in ill-advised directions, and risk-taking is frequently a part of it. And I think literally you can, one of the interesting ways we're thinking about this and actually doing studies of it at UCSD is that in mania, we all have a mechanism in our brains that weighs risk and reward. What are the odds if I run across this field that I'll be able to catch that rabbit versus being eaten by that tiger? And and literally, it's very interesting, the regions of the brain that have been implicated in this assessment actually involve mapping the internal organs. So internal organ sensation comes to this part of the brain called the insula. So it literally is probably developed by weighing the risk that you would be eviscerated, that you'd be killed, have your organs exposed to the world in the process of doing something versus being successful and rewarded. But this risk-reward mechanism in mania is distorted so that people overvalue the reward and undervalue the risk, and in depression it's the other way around. We also look for functional impairment as part of mania. If you had all of this but just had a little bit of it, and you needed only 5 hours of sleep a night, you had a little more energy than most people, you were charming, talkative, charismatic, Well, what would that be like? What do you think?
Great.
It'd be great. Where can I get it? That's what all my patients say. I want you to get me. Remember that spot, doc, where I was just, a little above? I want to go back there. Unfortunately, that's been, though a very popular request, it's difficult to stay there. And that really is the essence of bipolar disorder, is that people don't stay in any one place. Their mood is changing back and forth. So we distinguish mania from hypomania by way of several things, all of which are related to severity and functional impairment. And basically it's mania if you become psychotic and hear voices or see things, if you have to be hospitalized, or if you're functionally impaired, unable to go to work, get fired, get picked up by the police, etc. Less than that, again, if you have just a little bit of this, it's that hypomania that's very attractive. And it's, as we'll talk about in the next slide, it's people who only have hypomanias are in the category called bipolar type 2. In depression, the opposite, low mood, sadness, loss of interest or pleasure, what we term anhedonia, from the word derived from the same root as hedonism, their inability to experience pleasure. Change in appetite or weight, frequently losing weight and losing appetite, though sometimes the reverse. And insomnia or hypersomnia, unable to get to sleep or sleeping too much. And for many people with bipolar disorder, they have the mania where they don't sleep very much, they crash into depression, and then they're sleeping 12, 16 hours a day for a period of time until they recover. Fatigue, feelings of worthlessness, impaired memory or concentration can be so prominent in some people, particularly elderly people, where sometimes dementia is the initial diagnosis until you get to know the person better and understand what's really going on. This is how significant the thinking, memory, and concentration impairment can be. Most prominent on the list, of course, is suicidality. If left untreated, there's about a 17% fatality rate of suicide in bipolar disorder. But fortunately, we have some pretty good treatments, and some of them are very effective at reducing that suicidal risk. And then clinically significant distress or impairment. Depending on how you define it, as we'll see on the next slide, it's about 1 to 5% of the world's population, and remarkably consistently distributed around the world. There are a few places that are different. There's a South Pacific island of Palau, where allegedly there is no bipolar disorder. And then there is a valley in the middle of Hungary where the rate is about 3 to 4 times the rest of the world. But besides these few little pockets, it's remarkably consistent, which suggests that as a trait from a genetic standpoint, that these features of bipolar disorder have been in our population of humans for a long, long time, which is interesting to think about as well. So this is sort of what I call the taxonomy of mood disorders, divided into those who have unipolar depression and bipolar disorder. Now you first might ask, where is unipolar mania? That's a very logical question. It does occur, but it's pretty uncommon. And so it really hasn't even been in our diagnostic system until recently. In the next release of this, called DSM-5, due out next year, unipolar mania will be included. The people with depression may have just a single episode of depression, but when I went to school, I was taught that about half of people fell into that category. But as we study depression more, we've come to realize that even unipolar depression is really a recurring and relapsing illness. And it's that second line with the, let me explain here a little bit. These are the dashed lines are sort of the nebulous realm of normal. But this latter, of course, here of being recurring and relapsing, sorry, I didn't explain this very well. This is the lifespan over decades. Above the dashed lines is mania, and below the dashed lines is depression. The dashed lines is where you roughly say you're within the normal range. So this recurring and relapsing pattern seems to be much more common, and the number of people that have just 1 or 2 episodes is less common. Then in the bipolar category, we have people who have severe manias and may require hospitalization, full mania, crash into depression. This is what we call bipolar type 1. And then individuals with bipolar 2 have all the symptoms of mania, but just less severe. So instead of getting picked up by the police or getting fired from their jobs, they are instead getting commendations and raises because they're working 16 hours a day, and they're enormously productive and creative and are basically performing better than the average employee. During that period of time, however, that's followed by a period of depression where they may be relatively non-productive. Cyclothymia refers to people who cycle very regularly and but a sort of low amplitude but high frequency. They're constantly cycling up and down anywhere from every couple of days. The highs are relatively mild, the lows are mild, but what's impairing about this illness is that they're never normal. They don't know. It's the defining question is when you ask them, when was the last time you felt normal? And they look at you kind of funny and go, what do you mean by normal? Exactly. Because they're never there, and I think this is the impairing part. Now, also, this category, recently defined by one of my colleagues, Hagopa Kiskal at UCSD, was defined when they studied a set of people with depression. And they found that amongst those people with depression, there were some individuals whose sort of baseline operating pattern was a little higher than normal. That their normal baseline was to need only 5 or 6 hours of sleep a night, they have more energy, work harder, etc. But if you identified out of a group of all the people who had depression that they studied, and you pulled out those who had this sort of what we call hyperthymic or slightly manic baseline, that these individuals really belonged more in the bipolar category. They tended to have family members with bipolar disorder. They tended to have spontaneous manias in the future or become hypomanic in response to antidepressants. Yeah?
Would you say when someone's been put into a category, they stay in that category for the rest of their lives, or they're able to kind of shift up and down that classification?
These are pretty stable categories. I mean, nothing is sort of fixed in this business, but I think once you establish this bipolar 2 pattern, people tend to stay with that. Now, what we'll see sometimes is somebody who basically has a bipolar 2 pattern where they're hypomanic for a couple of days, once a month, and that's like their pattern for years and years and years. But then something happens to them, a severe stress, or they do methamphetamine, or they just have bad luck and they get picked up by the police. Then they may have a more severe episode, but basically these patterns tend to be fairly stable. Okay? So in treating bipolar disorder, here's kind of I want to put together sort of a list of the things we would like, our wish list for bipolar disorder. And one of them is medications that work better. We have medications that work reasonably well, and most people will go through a series of different trials, but eventually find something that really sort of fits them like a lock-and key, hopefully. But on the other hand, there's some folks who just, you run them through every medication and nothing really completely controls or manages them. So what we really need is like new medication. And my guess about some of these individuals is that their bipolar disorder may be different, that the biology of what's going on in their brains may be different, and it's not modulated by any of our existing medications. So a better understanding of the causes of bipolar disorder might help us understand those novel causes and mechanisms where we could develop new kinds of drugs that would help these individuals. The other is selection of medications. I think that unfortunately most patients with bipolar disorder have to go through a lengthy trial and error process. We have a lot of good medications that work very well, but we don't have any really good way to size you up and decide you're going to do well on Depakote, you're going to do well on lithium. So as a result, people wind up going through a series of trials, and these trials typically take 3 or 4 months at a pop. So you can have literally 3 or 4 years consumed in trials of different medications. Frequently at the end of that, somebody will hit the right med and they do great, but I'm always thinking, wow, what if there's just some way we could tell what that right med was to start with and skip all of these other meds in between would be very valuable. So this I'm going to talk about in a little more detail. We are interested in using, as I mentioned, genetics to try and predict response. So I'll tell you a little bit about those results in a minute. And then diagnosis, I think in 2 ways. One is a need for an earlier way of diagnosing. Right now, there was a study done a couple of years ago, actually, that used the DBSA, Depression Bipolar Support Alliance website to collect information, and found that out of 500 patients that on average, they went 7 years from initial onset of symptoms to an accurate diagnosis. And during this period of time, they received like 3 or 4 different diagnoses that were incorrect. Many of them may have been treated, either not treated or treated incorrectly. And being treated incorrectly in bipolar disorder can not just mean you aren't getting well as fast as possible, you might actually be getting worse because of the treatment, in that for some patients, being treated if the doctor thinks you have unipolar depression and puts you on an antidepressant, it may actually make some bipolar patients worse. And then also, I think sort of theoretically that we right now are, the brain is complicated and difficult to get at, as I'll illustrate in a minute. And so our understanding of the causes of these illnesses is relatively rudimentary at this stage. We know a lot, but we need to know a lot more. And our diagnoses are based on presentation and symptoms, and really defined by the question you ask, and that is which of these groups of symptoms sort of travel together in an individual over their whole life. And that's how we've built these diagnoses. But what do we really want in a diagnosis? We want a diagnosis I want a diagnosis in psychiatry to be like pneumonia. This person has a Pseudomonas pneumonia that's responsive to ampicillin and, or something like this. In other words, a diagnosis that really is defined by the cause of the disorder rather than simply its presentation. So in many ways, the diagnoses that we use now are more like syndromes. It's more like you would say 300 years ago, jaundice would be a diagnosis. But now we know there's dozens of different causes of jaundice, and each one of them may have a different treatment. So in that way, diagnosis should reflect the mechanism and cause of the illness, and that in turn should point to a specific kind of treatment. And I'm convinced we'll get to that point. I've just got back from a symposium. Our Institute for Genomic Medicine this weekend had a symposium, and they talked about cancer treatment, and this stuff is amazing nowadays, of sequencing your genome and finding the mutations in your cancer and defining a specific chemotherapy personalized for you. We need something analogous to that in psychiatry, and we're going to get there. But how do we get there? So, how to study brain disorders? Well, this is the problem we've suffered with for over 100 years. And that is that, as they say, we only want a small piece of your brain, Mr. Simpson, you'll never miss it. But brain tissue is hard to get at is the problem. And let's see, in Homer's case, there's not a whole lot of it to spare up here, is there? So nobody is or should be willing to spare any of their brain. And that makes it very difficult because we can't get at the fundamental organ of interest in order to study it. That and the fact that this is far and away the most complicated organ in the body. So trying to study something encased in bone that we can't get at directly, this is a lot of the reason why psychiatric research is perhaps not as sophisticated as cancer in a way. In cancer, I was talking to cancer docs over the weekend. I said, you guys are so lucky. Your patients are eager to get rid of their tumors. They're eager to get rid of the tissue that you want to study. You can have plenty of it, but people feel differently about their brains. So that's been the problem, and what I'm going to talk about now is how I think the exciting thing is that stem cells provide an amazing tool to get at how brains work and at brain tissue. So stem cells, very simply put, are cells that can become any other kind of cell. So after we all began as fertilized eggs and quickly turned into a whole bunch of stem cells that then go on and they have the capability to become any kind of cell, and they divide out in this fashion. A stem cell can become a bone cell, a immune cell. What we're interested in here is a brain cell, muscles, etc. Every different possible cell can be derived from a stem cell. So this has enormous implications and was sought after for really a couple of decades until initially it was thought that stem cells were very unusual or seen only in tissues that turned over very rapidly, like your bone marrow or the lining of your intestines. But nobody had any idea that there would be brain stem cells still in your brain until about a decade ago they were discovered, primarily by one of my collaborators, Rusty Gage, down at the Salk Institute. So they're critical in development, but what's been found more recently is that they're also instrumental in regeneration and repair in adulthood. So once you get to adulthood, you're kind of done building out the brain, but you need to keep a few stem cells around for maintenance and repair, and that's really how it kind of works. So there are stem cells at very low levels in certain parts of your brain. If something happens to you in potential, they would have the ability to grow out. And obviously this doesn't happen very much in the brain, but in other tissues happens quite a bit. They can come from embryos, as you may be aware, and this has some societal and ethical complications that have made them a little difficult to work with. What I'm going to focus on today is a new technology that's really different from embryonic stem cells, called induced pluripotent stem cells. Now, pluripotent just means they can become many things. And stem cell really just means at the top of these tree diagrams of which cell becomes which other kind of cell, they're at the stem, at the very top. That's all that means. So and this reflects a fantastic discovery by a Japanese group about 5 or 6 years ago, I believe, where they found that they could turn a fully differentiated cell. So we start out as stem cells, and then your cells slowly become other cells, and that process is called differentiation. So the amazing thing was that they found that they could take skin cells from an adult and treat them with 4 different genes that operated like a switch. And when you put these 4 genes into a cell, it flips that switch and turns that cell into a stem cell. It suddenly changes shape. It no longer looks like a skin cell. It becomes cells under the microscope tend to get kind of spread out and assume certain shapes as they become differentiated and assume their function. Stem cells are kind of round and amorphous and look like they could become anything. So they literally change shape. And the amazing discovery in this is really, who would have imagined that this was like operated in such a simple fashion, and that flipping a switch through using these 4 genes would completely change what this cell was doing. And that's been really an amazing discovery. So this is like the level of excitement in biomedicine about this kind of technology is hard to adequately describe. I mean, you see it in the paper a lot, but you should go to scientific meetings. I mean, it's just like all a Twitter with the possibilities. But for us, the exciting thing is that we can take a skin biopsy, a little piece of your butt usually, and then treat it with these 4 factors to turn it into stem cells, and then treat those stem cells with the appropriate growth factors and turn them into any other kind of tissue. What we are of greatest interest in is neurons. So we can actually know how to do this now, to take a skin biopsy, grow it in the lab, treat it with these factors, turn it into a stem cell, and then differentiate it into a neuron. So I can take a piece of skin from a patient who has bipolar disorder, and in a couple of months, I'll have living neurons in a petri dish from that specific patient. So that's just unbelievable. I mean, it's really enormously exciting as a tool. And I haven't heard anybody else use this word, but I've used the word cellular alchemy, just as the old alchemists who in the Middle Ages wanted to turn lead into gold. Well, this is turning skin into brain is just unbelievable. I mean, it was never imagined that this would be possible until 5 or 6 years ago, and now this incredible area has taken off. So this gives us a huge tool to study in the lab, and even Sigmund Freud down there in the corner would be amazed. After all, he did start his career as a neurologist, studying with the famous Spanish pathologist Cajal, and wanted to figure out how the brain worked. But after a few years of slogging away in the lab, he decided the state of the science wasn't really quite ready for that yet, so he moved on to invent psychoanalysis. So there's many, many different ways in which we can use a tool like this. What I've sort of illustrated are the different levels of analysis. At one level is the gene, and again, I'm so used to giving a genetics talk, and I have well, I may have some of these slides in here a little later, but probably studies of identical twins suggest that somewhere between 60 to 80% what causes bipolar disorder is genetic and inherited. So that's, we know that genes are very powerful. The genes in turn, some difference in the genes will result in some difference in the way neurons function, which in turn changes the way brains function, and in turn changes behavior. And we can analyze that behavior in people by asking them stuff, or in rats by watching what they do. But right now, we don't really have access to living tissue to study. We can get your DNA and we can compare it to your diagnosis. We can get your DNA and compare it to brain, but there we're limited to post-mortem brain, which is very, obviously, that's a very scarce resource. And it's very difficult to study for a lot of different kinds of reasons. So though a lot of good data has come from this, it's been limited largely by, again, the difficulty of getting at the tissue of relevance. We can make mice that have altered genes in them and see what they do. But what we don't have an ability to is to look at the neuron itself and see what's wrong. If the genes are, if there's an abnormality in the genes, then there's likely some abnormality in the way that neuron works. Neurons are basically entirely function by way of telling each other things or signaling through connections called synapses. If we had those cells in the dish, we might be able to see that they don't grow out right. They don't make the right kinds of connections. They don't fire properly. They don't signal. We get some better clue. And then what I'm really excited about is the ability to connect these two things with each other, genes and neurons. We may find a gene that we think is involved. We find a mutation in that gene, but we're not sure. It would be invaluable to get the tissue from an individual who has that mutation and then study to see, does it affect the electrophysiology, the electrical signaling of the cell? Does it affect the chemical signaling of the cell? And I'll show you just a little example of this. So, as I'm talking about, the cellular phenotype is a genetic term and corresponds to genotype is what your genes are. Phenotype is what you are, what you're like. So if I have a gene to make my eyes hazel, then the phenotype is the color of the eyes. So we'd like to be able to, as I was just saying, see what differences in neurons and their functioning is between people at risk for bipolar disorder and those who aren't. And can we correlate that with clinical features? Can we correlate that with DNA changes? And I think this kind of understanding may lead better to that biology-based system where we understand certain individuals are more likely to have this kind of illness because they have certain kind of signaling system wrong in their cells. And again, as I mentioned, the functional effect of gene variants. Right now, just to illustrate from our own work, we identified a family a number of, like 15 years ago, that was unique for having a rare kidney disorder being transmitted through the illness along with bipolar disorder. So everybody who had bipolar disorder also had the kidney disorder. Now that, from a genetic standpoint, is very exciting because of the prospect that there might be a big deletion of chromosome that takes out a kidney gene and a bipolar gene, and by defining that deletion of chromosome, you find both genes. Now, from the family's perspective, it's awful, and this has just devastated this family. In this case, really, the kidney disease being worse than the bipolar disorder. We've gone through literally a 10, 15-year process in multiple different technologies, and now we think we have identified the gene and the mutation in that gene that causes bipolar disorder in this family. But we need to prove it, and one way we need to prove it is show that the mutation in the gene causes that gene to malfunction in some way. And that's what's been very difficult to do. So last week we met with a lady who's going to give us a little bit of skin, and then in probably about 4 to 6 months we'll have her neurons in a dish, and we'll be able to look to see if they malfunction in the way that we expect them to. So there's been relatively little work in bipolar disorder to date. We're doing some of this work now, and I'll tell you a little bit about what we're doing. But there's really just 1 or 2 papers out right now, and one of the biggest papers has to do with schizophrenia rather than bipolar disorder. But we are working with these folks in this group. This is Kristen Bernand and Rusty Gage at the SALT. and hope that we may be able to do studies like this in the near future. So what did they do? They took a set of about 6 people with schizophrenia and 6 controls and made these iPS cells, stem cells, and then differentiated them into neurons. So they had neurons from people with schizophrenia and neurons from people who didn't have schizophrenia. And this is what this, and essentially, to make a long story short, they injected a part of the brain with a virus that was labeled. And then if signals were transmitted through the neurons correctly, it lights up in red like this. When signals are not being transmitted down the sort of the wiring of the brain as the part of the neuron called the axon, when this wiring isn't working correctly, there's not much red, meaning that signal didn't really get to its target. So this is consistent with there being a lower level of connections in the brain in people with schizophrenia. They went about it another way and essentially just looked at the cells in detail under the microscope. Neurons kind of spread out, and they make these little things sticking out called neurites, and basically showed that there were fewer neurites in the neurons from people with schizophrenia than controls, and that when they treated the cells with an antipsychotic, in this case one called loxapine that would be used to treat schizophrenia, that this began to correct itself. So that's really pretty striking and suggests that a fundamental abnormality in schizophrenia may be the ability of cells to connect with each other. And that's, this is of course the main thing neurons do, so that's a major, major problem. Now we don't know if this is what happens in bipolar disorder or not. We have learned in the last decade that bipolar disorder and schizophrenia probably share a lot more in common than we used to think, and that the genetic studies suggest that about half the genes are shared between these two disorders. So that's very interesting. Now, in another study, a group at Harvard and MIT are using stem cells to try and identify new drugs for treating bipolar disorder. So lithium is I'll go into lithium in more detail in a minute. It's a complicated story. People have been trying for decades to figure out how it works. And our problem right now is that we know too much about how it works. It inhibits all kinds of things in the cell. We don't know which of those are really the important things. But one thing that's, there's one pathway that's inhibited by lithium that's sort of a major candidate, and that's what these guys went about doing. So they modified these stem cell derived neurons so as to be able to measure the degree to which a drug was inhibiting the same pathways in the cell that lithium does. And this is just a whole bunch of compounds that they screened looking for new drugs, and they found some that didn't work so well. But here's 45 compounds that led to a significant change in that pathway and represent candidates for treatment of bipolar disorder. So this doesn't really, this is using a stem cell as essentially anybody's neuron would do in this case, but it's possible to really do the same kind of project on stem cells from people with bipolar disorder and see how that works. And even further down the road, though I don't think we'd be screening thousands of compounds for an individual patient, but it might be possible to take a stem cell from you and determine how your cell responds to different drugs and pick the best medications for you based on that. So that's, I suspect that that probably will never really happen because it's too complicated and takes too long, but we will use that kind of design to probably come up with genetic tests that might substitute for it. But who can say what's going to happen? It's so unpredictable in this business. It might turn out that this becomes a routine clinical procedure. All of this has been categorized in the general title of personalized medicine, and that just simply means instead of giving the same drug to everybody who has a diagnosis, that we try and figure out which drug or treatment each individual is most likely to respond to. The whole idea that people with bipolar disorder come into the office and have to go through trials of 8 different medications over 2 years before you find something that works is an indication of the need for personalized medicine. And the fact that everybody's different. And I think there's very good reason to believe that probably you can get to bipolar disorder by multiple different pathways in your brain, multiple different pathways that can function abnormally and lead to risk to mania. We need to be able to figure out what those are and tailor the appropriate treatment to the individual. So this has sort of been fueled by the Human Genome Project. It's one of the great promises out of the Human Genome Project. And my group, as well as many others around the world, are working really hard to try and make this happen. So that's my segue to lithium, and talk a little bit about pharmacogenetics, and then we'll get back around to how we're using stem cells. So lithium, I'm sure you're all familiar with. It was the first in a category of mood-stabilizing medications, and there's just a fascinating story behind its discovery. I don't know if you guys know this, but a guy named John Cade in the 30s and 40s in Australia was studying lithium. He was actually interested in a different compound that he thought was causing schizophrenia. And he was simply using lithium to precipitate that other compound out of solution as a salt. So just like you have sodium chloride, your lithium chloride is just using it. He didn't think lithium was the relevant thing. So he wound up giving this concoction to patients to see if it would be helpful, and lo and behold, they got better. Only later he figured out that one, it wasn't the other compound, it was the lithium that was doing it. And two, that these folks that got better weren't really patients with schizophrenia, they were ones who had bipolar disorder. So there had not really been any medication like this prior to this. And the striking thing about it is lithium is you don't get any simpler than this. It's just an atom. It's a single element. It's not even a compound. It's just an element. But this has the strongest, but lithium had been around for a long time. There were, there were lithium waters you could drink or bathe in to calm your nerves. And then there's also this interesting story of how epidemiological studies have been done and shown that in areas around the country where there's a high concentration of lithium in the drinking water naturally occurring, there's a lower rate of bipolar disorder. So that's kind of an interesting observation. But this came into clinical use in the US in about the 70s and has been sort of the gold standard for treatment ever since. Now, my sort of personal campaign as an educator and a doctor over at UCSD is to teach my doctors to use lithium. I've been known to threaten them that they wouldn't graduate if they didn't put somebody on lithium, because I feel like it's a fantastic drug, and it's gotten the reputation of being, what do people say, too strong, too heavy. I'm not so sick as to need lithium, or that it has terrible side effects, etc. And there certainly are side effects like any medication, and there are serious risks to watch out for, but this is really not much more difficult than any other medication to manage. It really still has the strongest evidence for efficacy. It works the best. There have been some, my concern is that through advertising from pharmaceutical companies, it's almost dropped out of use and been taken over by other medications. But yet, there's been recent studies that still show it's the most effective of any of these medications. And really importantly, it has the strongest evidence for reduction of suicide risk. It's the most powerful anti-suicide treatment that we have. It is literally dirt cheap, comes out of the ground. It costs like a penny a day to treat somebody with a lithium. Does have side effects. I put weight gain up there, but honestly, we've studied like about 150 people in our study of lithium right now, and we've seen very little weight gain. Apnea, it'll make you drink a lot of water, make you pee a lot. It may affect your thyroid, which is a concern, but can be managed by watching your blood levels. And more seriously, it can have permanent damage to your kidneys, but typically only after decades. And with regular monitoring, you can pick that up well before it becomes serious. So I think that now the thing that really motivates me is that there's a subset of people with bipolar disorder who have a fantastic response to lithium. You put them on lithium and their illness is like gone. And as soon and as long as they stay on it, they'll not have an episode at all. And this is maybe 10–20% of patients. But there's increasing evidence to suggest that they may not get that fantastic response unless they try lithium early on when they're young, early on in the course of their lifetime course of their illness. So it's my belief that every person with bipolar disorder deserves a trial of lithium to see if they fall into that category. And then you'll know. Otherwise, I think I just feel like it's a shame that there are probably some people out there who might get that fantastic response, but have never been tried on. Yes, sir?
Is it critically important that you have that lithium level in the blood for efficacy?
You need to have the lithium in the blood.
But at that level 4 to 0.7 to 1.1 or some, whatever it is?
This is kind of going back and forth. And a number of years ago, there was an argument it didn't need to be quite as high, and then other data came back in and said it really ought to be 1.0. I think that statistically, considering everybody on average, you'd like to keep somebody in the 0.8 to 1.0 range. But everybody's different. So really, what matters is not what the blood level says, what matters is what the patient says. And if the patient's doing great and their level is 0.4, I don't mess with it. I stick with what works. Because really, you take it to a higher level and it might not be able to tolerate it. So I think this all has to be sort of, so statistically, yes, the answer to your question is yes, but everything needs to be personalized. Let's see, okay, so I've said all of that. So there's, and part of what suggests that the interesting idea that's been in the field for a while is that these excellent lithium responders really have a different kind of illness. Some people, some experts in the area would argue that it's that only these really excellent lithium responders have what is real bipolar disorder. I think that's defining things a little narrowly. But certainly there's an argument that they may be different. And this is some of that argument, that people who respond well to lithium have a different set of clinical features. They tend to be more likely to have euphoric or elated mania instead of irritable mania, or as I gave a talk to the DBSA about a month or so ago, and the one person was trying to explain it to the other in the audience says, there's happy mania and there's pissed off mania. He's talking about pissed off mania. So it's the happy mania that usually responds best to lithium. And the second predictor is that there's a strong family history of bipolar disorder. Those are really, of the things on this list, those two are really the most powerful. A good response in the first 6 to 12 months predicts that they'll have an ongoing good response. And then a family history of lithium response, which I'll show you in a second. And then kind of, they tend to have lack of other features. They don't have as high a rate of substance abuse or alcoholism or anxiety disorders. They'll also tend to have mania followed by depression followed by euthymia, as opposed to depression followed by mania as sort of a episode order. And the depressions tend to be more severe and have what's termed, a set of features termed melancholia. This pattern is different from that of some of the other drugs that we use. Family history doesn't necessarily predict very well for some of these other medications, or rather for lamotrigine, what you see in the families is more anxiety and depression than bipolar disorder. Mixed states where people have symptoms of mania and depression simultaneously are really don't respond very well to lithium. They're best treated with some of the epilepsy medications like valproate or carbamazepine, Depakote and Tegretol, and then rapid cycling may do better with some of the anticonvulsants as opposed to lithium. But lithium is also one of the best in terms of managing both mania and depression equally well. So there's a little bit of a difference in pattern. Lamotrigine is very effective for the depressed phase of bipolar disorder, relatively ineffective for the manic phase. So these drugs have different patterns, and the distinctiveness of the clinical pattern for lithium, again, is another piece of evidence suggesting that these individuals are different in some way. So, wife was telling me this slide is too complicated. It probably is. But so the question addressed here is, does lithium response run in families? So what they did, this is Paul Groff and a group in Canada, looked at a scale which scores how well someone's responding to lithium, named after Martin Alda. And then they had a group of people who are really good lithium responders, and then they identified those who had relatives with bipolar disorder who had been treated with lithium, and then asked the question of those relatives of people who had a good lithium response, how well did they respond to lithium? And in general, they responded to lithium pretty well. This is just two different ways of scoring it, but both of them are elevated above the comparison group, which is just kind of whoever walks into the clinic with bipolar disorder. So if you have bipolar disorder and have a relative who did well on lithium, you are more likely to do well on lithium than if you didn't. Again, suggesting a genetic basis to this. So again, what I've been building up to is the idea that is lithium-responsive bipolar disorder, a genetically distinct form of illness. So all forms of bipolar disorder, lithium-responsive represents a subset of that and potentially a different mechanism of illness. And the group in Canada that I was referring to took this as a number of years ago in an approach to try and see what if we just try and find genes for bipolar disorder in people who respond really well to lithium. And not to get technical, but they were modestly successful in doing this, in identifying a possible gene on chromosome 15. So as we think about the genes that modulate drug response, there are several different ways to think about it. What kinds of genes are you going to find? And in the medicine business, when you say pharmacogenetics, most people think about what's called bioavailability. You take a drug, your body immediately recognizes it as a deadly poison and tries to get rid of it. Because this is what your body does. When it recognizes compounds that aren't supposed to be there, it tries to get rid of them. Because in the old days, you were out there eating berries or eating all kinds of stuff. Who knows what you got hold of? So your liver functions to detoxify poisons out of your blood. They're usually stuff that you eat. But it has a whole range of different enzymes that chew up foreign substances, and this is what chews up the drugs. So the amount of drug in your blood depends, one, on how much you take, but two, it also depends on how fast your liver chews it up. So this doesn't apply to lithium. Lithium is just an atom. It's peed out, and the elimination is entirely regulated by your kidneys. So, so this has been historically what's been considered the major feature of pharmacogenetics. But in this case, it definitely isn't, because we monitor how much of it is in your blood because we get blood levels on it all the time. And some people may absorb it. Some people may require lower dose to get to that 1.0. Other people may require a higher dose. But we know where it is in the blood. So of those other genes, what are they? Some of them may be genes that also cause a susceptibility to illness. In other words, if a gene both modulates the action of a drug and exposes you to risk for having bipolar disorder, then what do you really have? You have a gene that predisposes you to a lithium-responsive form of illness. So this is linking the cause of the illness and the response to treatment together to each other. Then there may be other genes that don't really make you more likely to have bipolar disorder. They aren't causing your bipolar disorder, but they are modulating how well the drug works. Is it due to the genetic variation in the targets where lithium was working may make it work better or less, but those genes may not influence whether you get the illness or not. So this is simply illustrating the same point in a different way, and that is saying under the hood, if you will, there are several different ways you can get to bipolar disorder. We come into this world with a set of genes, and some people may be predisposed, may have abnormalities in different kinds of signaling pathways in their neurons. Then they may respond to different medications, or that predisposition and interacts with environment and other weird things. So I don't have time to get into what that means, but it's a very weird story. And then, but this person may respond to lithium because the pathway that because their illness is caused by disturbances in a pathway in their neurons that is modulated by lithium. Then other people may not respond to that, but may respond to other drugs because their illness is caused by abnormalities in pathways that aren't affected by lithium, but are instead modulated by other drugs. So this is kind of the theoretical underpinnings of how we're thinking about going about understanding, finding genes that predict response to medications. And the thing that got me really excited about this is I could see that once you found genes that predicted response to lithium-1, and most importantly, you have really something that could possibly relatively quickly be taken to the clinic, and that's exciting, and trim those 4 years of med trials down to maybe 1. But also, you could use the medications as a probe to divvy out different causes of the illness. So that if we, just like we were talking about before, where Tarecki and Aldo were studying lithium-responsive families to try and identify genes, this is an example of how we go about sort of divvying it out, simplifying the problem. Now, to make a long story short, what we did was we've taken about, started out with about 200, now we have about 400 patients who've been treated with lithium in the past. And we took a very simple approach and just asked them, have you ever been on lithium? And how did you do? How did it work for you? And this is, so we obtained information about their response by history, or what would be termed a retrospective assessment. So that's, in many ways, the information may not be quite as good, but it did get us some subjects pretty quickly. And we found this remarkable thing. I'll talk about this a little more in a minute. But this gene, there's lots of reasons. This gene called INTRAC2, which I'll tell you about in a second, there's many reasons to believe that its function is modulated by lithium. So we tested it to see if variation in this gene was in fact correlated with lithium response. And this is what we found. So if this is the response rate, so up here means a really good response, down here means a really poor response. And then these different bars represent the 3 different possible genotypes. So at a single, at a given gene is made up of DNA that's made up of bases. Those that are like letters in an alphabet, or letters making up a word, is really a very apt analogy for how DNA operates. So since you have 2 copies of everything, one from your mom and one from your dad, everybody has 2 different copies of the same DNA. Those letters can be an A, C, T, or G, which just stands for different chemicals that serve as letters in the genetic alphabet. So if someone had so you can have a TT, TC, or CC, or the 3 different possible combinations of 2 letters. So here we found that basically differences in the DNA sequence here, if the person had dysphoric mania or irritable mania, they didn't respond as well to lithium in general, which is what we would have predicted, but also variation in this gene doesn't really make any difference. But over here, if you have no copies of the T allele and you have euphoric mania, then your ability to respond to lithium is not that much different from these folks. But 1 copy of the T allele gets better, 2 copies of the T allele takes you up into the 80% response rate, which is really quite striking. So I think that this is very exciting because, 1, finding something that was associated with response that made a really interesting biological story, but also the fact that we saw it only it interacted with the clinical presentation. We saw this association only in that subset of patients who would have been predicted based on their clinical presentation to respond to lithium. So that's pretty interesting. Now we went on to, well, first of all, what is INTRAC2? And not to get too technical here for you, this codes for a gene called TRAC-B. That's a receptor for growth factors. So when it's been found that when we treat animals with antidepressants or lithium, that a growth factor called brain-derived neurotrophic factor is increased in expression. If you block that increase in expression, these drugs don't work. So we believe that this increased release of BDNF is really necessary, but probably not sufficient for these drugs to work. Actually, I think what the theory is that these drugs turn on expression of these growth factors that then tell your neurons to get ready to remodel, that they're going to you're going to rewire the connections between the different neurons, and that rewiring may be part of, or maybe the main part of what's necessary to heal and to get better. So we similarly know, yes?
So is there a suggestion that lithium exposure then does cause additional contacts to be made, additional rewiring going on? Is there any evidence for that, like in a preclinical model?
That's an excellent question. I'm not sure I know the answer to that.
It's hard to do in a patient.
I read that there's an article I read maybe just about 6 months ago that said lithium could, basically they're studying it for Alzheimer's disease.
Lithium does lots of stuff. And one of the things that it does is protect your neurons. It has what's called a neuroprotective effect. And that is, some of this comes out of studies of stroke, where at the margins of a stroke you have neurons that are ill but not dead yet. And one of the key factors in managing a stroke is to really get to somebody as fast as possible so as to save those neurons that haven't died yet. And it's been found that lithium has a protective effect in these kinds of models. And there's a long theory about exactly how that works. But I think that's been some of the idea of using this in Alzheimer's. This is sort of that this may have a general protective effect on neurons. This is probably mediated via BDNF and other related growth factors. That again, when you're when you're in development in your mama's belly, these things are turning on and telling your brain how to grow out. But as an adult, they may be more involved in repair and maintenance. So this may be telling your brain that it's time to remodel and repair and maintain things. Another approach to it is to say that stress releases steroid hormones in our system, and those steroid hormones have been shown to literally kill off neurons. And the antidepressants and lithium turn on BDNF and may, in part, help literally grow some of those neurons back, or at least make them healthier. And the other interesting thing about it is that as you look at these are just pathways of different proteins that modulate other proteins in your cells, but many of the things that this receptor, this is your track B or in track 2 right there, many of the things that it's connected to are modulated by lithium and have been candidates for being the primary mechanism of action of lithium. So that's also what makes this interesting. Now, we did a study a few years ago where we took 1,000 people with bipolar disorder and 1,000 controls, and we looked at 1 million different DNA markers in these individuals. This has been termed Genomewide association. We looked to see was there any association to any genes. And this is a whole separate talk, but I just wanted to take a long time to explain all of this. But here's the point, and that is in track 2. So what this plot is your chromosomes laid out end to end, 1, 2, 3, 4, 5, blah, 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 22. And then in this axis is essentially the evidence that this gene is causing bipolar disorder. So what we found right here, this dot, is in track 2. So that means that there's evidence that this not only predicts response to lithium, but it also may predispose people to bipolar disorder. So remember what we were talking about a little bit earlier, that if we saw that kind of pattern, that would be consistent with intract2 being a gene that predisposes you to a lithium-responsive form of bipolar disorder. Now, also of interest is in the last couple of years, recently a report came out that showed that variation in intract2 is also associated with risk for suicide attempts. So this is very interesting when you come full circle back to the fact that lithium is the best anti-suicide drug that we have, and it may be operating through this gene, and this variation in this gene exposes you to risk of suicide, really makes an interesting story, and also very interesting, seems to point to specific kinds of biological pathways that may particularly predispose someone to suicide. So that obviously is the most important thing we want to treat and get at, and is very exciting. Now we went on and, well, here's the bottom line, is I wanted to expand our retrospective sample and just get thousands of people and ask them if they've been on lithium. So, but I sent my grant in to not just one, but 2 granting agencies, and both committees came back and said it needed to be a prospective trial. So a prospective trial is what is usually done in pharmaceutical companies developing new compounds, where, so now what we're doing is we bring patients in, and we're doing this in 2 studies right now, one strictly of veterans at the VA hospital, and another one is a much larger study that I'll tell you more about in a second. Well, we bring patients in, and for about 3 or 4 months, we stabilize them on lithium. And this usually means starting them on lithium and then very slowly tapering them off other medications if they're on them. and then to see if within about 3 or 4 months we can get them stable and symptom-free on lithium alone. Now the key element is that the problems with the retrospective design are, one, people are trying to remember what happened 10 years ago, which is pretty sketchy for the best of us, and then, but also that people are generally on multiple medications. So someone may say, I was on lithium during this period of time and I was doing really well, but they left out the fact they were also on Zyprexa during that period of time or something else. So you don't know which drug is causing what. So this design enables us to get people on lithium alone. We're observing them while this is happening, and it should just generally be a much higher quality of data. It's just really hard to do. So we have about 120 patients in our study right now at the VA. In this analysis with 777, we found that basically this is the length of time. Here they're started on lithium, here they're on lithium alone, and then we follow them for 2 years to look for relapse. So, and then we look at time to relapse as sort of a quantitative measure of how well they did on lithium. And what we found, again, with the same gene, the same T, these individuals, so as you go through time, people drop out as they relapse, you lose subjects. This is how many subjects are still remaining. So if you're high up on the list here, this line indicates that people who had this TT genotype, same as in the other study, did better and had a lower relapse rate than people who had other genotypes.
Question?
Yeah.
What percentage of your initial sample were you able to get stable on lithium alone?
Well, that's an excellent question. We have about 20% that drop out for a variety of unrelated reasons. We get about half of them stabilized, about 30% who fail to respond, and then somewhere between about 40 to 50% who we get stabilized.
And you're looking just for the reappearance of a manic episode?
Or depression, either one. So this led in turn our VA study, then I took that and I leveraged it into a bigger NIH-funded study. So now we've expanded this. We recruited a bunch of other sites and in the US and in Norway and Canada. And it's a very similar study. We're just scaling it up. So we're doing pretty much the same thing I just told you. But now we're doing the same thing at all of these different sites, and we hope to get up to 700 patients in this design with 2-year follow-up. And then in the end, we initially designed this to do genotype 1 million markers and do genome-wide association. But the technology is moving so fast that I think that by the time the study is done, we will be able to sequence the entire genome of all of the people in the study, which is just an utterly amazing thing to do. I'll tell you how amazing, and that is when I started in this business a long time ago, I imagined that one day I'd see the entire human genome. Well, that's like a decade ago. And then I thought, well, this is really great. I'll probably get to do, no matter how hard this genetics of bipolar disorder problem is, because it seemed to be pretty hard, that one day in the distant future, we'd be able to sequence the entire genomes of a whole bunch of people. Now, at that time, that honestly seemed like a problem of such vast proportions, it was almost inconceivable. But we are doing it now. I thought at that time, well, that would be great. While I'm retired, I can watch those young kids do it. Now that would really be entertaining. But now, here we are doing it. So it just speaks to how fast this technology moves. It's just mind-boggling. It's been in the Human Genome Project. After a sort of a paper was written and literally kind of a challenge made to the community to develop the $1,000 genome. So, and this is likely going to be realized by the end of this year, where it'll be possible to have your entire genome sequenced for about $1,000. Yes, sir?
So, you talk about the technology moving so quickly, is it because we can sequence faster now than we used to be able to, making this all possible?
Yeah, basically. It's gone incredibly parallel. So the way these latest machines work is they basically built a big semiconductor chip, and on that chip they put 600 million pH meters. So this measures the acidity in a solution. And then let's see, how's it work? They wind up, there's a tricky method where they wind up putting a different little piece of DNA in all 600 million different wells. And then each time, and then they synthesize the DNA in the well, so each time a base is incorporated, it releases a proton and changes the pH, and they measure that. So it is just really totally, and the other cool thing is that in the world of genomics, where sequencing technology is what it's all about, the two companies in the world that are developing this, that are really at the lead, are both here in San Diego County. One's like about 3 miles from here, and one's up in Carlsbad. It is, it's just amazing. So if you want to learn more about it, go to www.lithium.ucsd.edu and you can learn more about our study. So this is basically the design we're talking about, where we stabilize patients. We observe them for a month after they've gotten down to lithium alone, just to make sure that when we removed that last other medication, they weren't going to relapse. And then we follow them for 2 years and watch people very carefully and monitor them very closely. If they fail lithium, they get crossed over to they get tried on valproate, a different medication, as a comparison group. Now we're collaborating with another group called Conlogen, and that's some of these Canadians and Germans. There's a whole international group. They have like, I don't know, I think there's probably 15 different countries represented in this sample. Everybody got together and put together all the subjects for whom they had information about lithium response. And they've now done genome-wide association with 1 million DNA markers on about 1,500 people on whom they had evidence for they had measures of their lithium response. So we're hoping to start by taking kind of what they find and then to see if we can replicate it in our sample is one approach. Now, back to stem cells. I told you I'd get back here. So this whole design, made us think that there would be a very cool application here. And this is the ability to have living neurons in the Petri dish from patients who did great on lithium and from those who didn't do well on lithium at all. And then can we compare those two types of cells to see, is there a difference in the way they grow neurites, in the way they connect to each other, in the way they signal, the way the electrical signals work. The other thing so that's sort of the cellular phenotype. How does the cell function? But then to take it a step lower to the DNA level, what we intend to do is treat these stem cell-derived neurons, both with and without lithium, in people who were good lithium responders and poor lithium responders. Then we look at all the expression, the function of all the genes in these 4 different types of cells. And we look to see which genes change their functional response to lithium in responders but not non-responders, or the other way around. But can we find genes that are affected by lithium that are different between these two groups? Then maybe we've found a gene that's really critically involved in both lithium's mechanism of action and the cause of bipolar disorder in these individuals. So now we've identified these genes that may be candidates. Well, remember, we're going to sequence the entire genome of all of these individuals. Then we go back and look in the genes that changed only in responders and look in their DNA and see what's different about the sequence in their DNA that may tell us why those genes changed in a different way. This will help us get to a point of understanding which genes may modulate response to lithium as well as expose someone to risk of lithium-responsive bipolar disorder. And also begin to find variants that could be included in a test. And we're actually, I've gotten very excited about this pharmacogenetics, and for all of these different reasons. And as part of that, I'm part of this group called Pharmacogenetics Research Network that's sponsored by NIH. And they are very motivated to try and promote implementation of pharmacogenetics. So how do we get doctors to actually use this in the clinic? Well, us psychiatrists, though we are incredibly stimulated by the prospect of a biological test, having never had one before in our careers, But a little bit, those practicing docs are a little bit intimidated to hear about DNA, which they dimly remember from 30 years ago when they were in med school. And the truth is the field changes so rapidly, whatever they were taught back then is probably only about 50% true. But so there's a lot of hurdles, systems that you have to get over. How do you get the DNA results in the medical record? Because it's not like an ordinary lab test. This never changes. Well, it really should be available all the time. It shouldn't just sort of get older and expire the way normal lab tests do. So a lot of different kinds of things. But one of the things we're trying to do, one of the things that's really needed to get doctors and hospitals and systems to adopt this is not just evidence that a gene is associated with response, but to take it to the next level and show that if you give doctors access to this kind of genetic information, then patients do better. So that's what we're just about to start at the VA right now. We're working with a local Pathway Genomics, a local company down in Sorrento Valley. We've developed a test panel, and then we will randomize patients to those whose doctors get the results of a pharmacogenetic test panel and those who don't. And that panel will say things like, this person is more likely to respond to lithium, less likely to respond to Depakote, more likely to respond to Prozac. They'll also say things like, don't treat them with Zoloft because they are unable to metabolize it and they're going to have a lot of side effects. Or it may, or even worse kinds of things. So it picks up a lot of things that may result in extra worse side effects or poor tolerance, as well as you may find somebody whose liver chews up the drug really quickly, and that might tell the doctor that this person may respond, but they're going to need to be on 2 to 4 times the normal dose in order to get the adequate blood levels. There's many different ways in which this can work, and hopefully in the next couple of months we'll start this, and in a year we'll see if patients whose doctors get this information actually do better. And that, if we're successful, hopefully will be the kind of evidence that will be necessary to begin to implement it, first possibly at the VA and then beyond. Didn't go over that, did I? Got distracted. So stem cells that we talked about can become any kind of cell, hence the term pluripotent. They can become many different things. Skin cells can be reprogrammed into stem cells, and what's termed induced pluripotent stem cells. These iPS cells can then be differentiated into neurons. And this will help us understand disease mechanism at the cellular level, potentially discover new drugs, and develop ways to predict drug response. So these are some of the many reasons that we're excited about this. And this is just a cast. It's a cast of characters involved in this. My wife, Susan Leckband, and wife and collaborator, as well as Anna De Medina, who runs the study here locally, Tanya, who's done much of the genotyping, my colleague, Hagopa Kiskil, who's been instrumental in thinking about bipolar disorder, and the whole team here. And then these are some of the other sites around the world that are involved in this study as well. And I always like to put this up here. This is I usually do this at scientific meetings. So I'm in Michigan in the wintertime. I like to put this up from San Diego. My wife is meaner still. We're going to Michigan next week. And she has a slide like this with a sunset. And then in the middle of it right here is a picture of her iPhone's weather that says San Diego, 85 degrees last week. That I thought that's just a little mean to you guys in Michigan. So that's my talk, and I'd be happy to answer any questions. Yes.